And that's why Mike was on.
Yeah.
So have you actually started recording yet?
Yeah, it's recording.
All right. Well then, hello everybody. For those of you who are from the outside, today is August 31st, 2017. I'm Dr. Newman, and this is at Einstein Neurology. This is from the Basic Neuroscience series. This year is Neurophysiology. Today's lecture is part of a macromolecular neurochemistry series, and this is the first of several lectures that will span the next 2 years. Our topic today is cell membranes. This is how do I make this go sort of full size? Maybe it's F5.
Down.
That, very good. All right. This topic is near and dear to my heart because this is what my PhD was in. And if you ask me where I live, where my brain is inside the cell membrane. And so my view of biology is from that of the membrane. And that may explain some of the bizarre things that I say. There's a lot to do in this lecture. We're going to very briefly talk on the functions of the cell membrane. Then we're going to go right to basic properties of lipids. This is about chemistry. And we're going to look at the forces that create stable bilayers based on lipids. And then we'll look at how proteins interact with them, all the way down to their functions. You could make this list yourself. The functions of cell membranes is, first of all, it separates the cell from the rest of the world. So that's probably its single most important thing. But it also separates the intracellular compartments as well. Through all of those membranes, whether the cell membrane, plasma membrane, or rather an intracellular compartment, the properties, the intrinsic properties of the bilayer that we'll talk about include a selective permeability, and therefore the ability to use pumps to generate electrochemical gradient. This is obviously of critical importance to the nervous system. But it also serves for sequestering of molecular species and the activities. They can be compartmentalized. It will participate in the control of cell shape, intercellular adhesions, and intercellular communications as well. All of these could not happen without a cell membrane. Okay. Quickly, some examples of intracellular compartments that this list is growing over the years. But some of the phylogenetically oldest include the chloroplast and the mitochondrion. We can also include the photoreceptor in the eukaryotic cell, the endoplasmic reticulum, the Golgi, the lysosome, peroxisomes, the nucleus itself, of course. And there are other proteasomes and other things that are being discovered as time goes by. Now, when we start at the beginning, I think you want to look at lipid properties. And we'll start with the simplest of all, which is an oil or fat. And from a chemical point of view, these are triacyl glycerols. That means that they're on a glycerol background, and I'll show you the structures in a minute. And then it has 3 fatty acid chains attached to the glycerol. So that's a triacyl glycerol. These things are completely hydrophobic. This is when you say that oil and water don't mix. This is what you're talking about. They have nothing in the least bit hydrophilic. There's nothing polar about them at all. And so they're just going to sequester themselves. They're going to adhere to one another and avoid water like the plague. And they will form a skim on the surface of the water. And that's when you look at oil on the street and that sort of thing, that's what you're looking at. Now, fatty acids, first of all, you have a single acyl chain, and it introduces the carboxyl group at one end. So it's not like the glycerol, which itself is nonpolar. Now you're putting a carboxyl group, and so that's going to have a negative charge. That's going to make that section hydrophilic. The tail remains hydrophobic, And so you end up with what's called an amphipathic molecule. It goes both ways, right? That's important. It's going to want to sequester its hydrophobic compartment, and it's going to want to expose the hydrophilic group to the water. So it's going to orient itself. A diacyl glycerol, as opposed to a triacyl glycerol, means that one of the fatty acids was taken off, and you have a hydroxyl group sticking out there. That is minimally hydrophilic. But the thing that this does is once you have the hydroxyl group, you can put something else on that hydroxyl group. And I'm going to show you pictures in a second. And the most common thing that is done is to put a phosphate group on that hydroxyl group. And that makes phosphatidic acid. And that is very, very polar. So let's take a look at some molecules here. Here's the glycerol. So this is glycerol over here. And this is a triacyl glycerol. Notice they're esterified. So this thing is more or less, if I broke this bond here, I would be creating a fatty acid. All I need to do is take that off that group, okay? So when the chains are all long and there are no cis bonds to disrupt the structure, they can all align with one another, and you end up with a fat. That is, what we mean by that is at room temperature, this thing is a semi-solid. But if you make the fatty acid chains with bends in them by putting cis bonds into them, like here, you can see this thing. This means that they don't pack. This thing, I assume you would have no problem seeing how this thing would pack and make a wax or fat, okay? This thing will not do this. This thing at room temperatures is going to stay liquid, and that's an oil. So the only difference between both fat and oil are triacyl glycerols. The only difference is that the fatty acid chains are bent, and so they can't anneal with one another. Here's that fatty acid. There's the carboxyl group, the single acyl chain, and the carboxyl group. That's what we said was a fatty acid. And this is a fatty acid micelle. This forms spontaneously. You take a bunch of these and put them into water, and this is what they will do. Unlike the lipid, the pure fat or the pure oil, which will just rise to the surface and do all that it can to stay away from the water, this thing doesn't mind the water because it can orient itself. Now, if you also had, if you think a little biologically for a minute, if you also had some oil in the water, so you had oil and water, okay, and that's all that you have, they won't mix. Put some of this in there, and basically you have a soap, because this thing is capable of putting its fatty acid chain in with the oil and orienting the carboxyl head group to the water, and so it can emulsify the oil, and you can wash your hands and get the oil off them with soap and water. And that's basically what you're doing. So these are typical, this is your typical soap. Notice also in this diagram that, sure, the carboxyl groups are all aligned out into the water, so they stay out of the hydrophobic center of the micelle. Notice also that at physiologic pH, they drop their proton, and so they're all negatively charged. So this thing is a lot of charge, right? And therefore, it will attract mobile cations, and sodium and potassium, but also calcium and magnesium will come to the surface of these things and bind to the negative charges of the carboxyl group. Okay? Right. So this is just a summary of some of the what I talked about before. This you saw before. This is a triacyl glycerol. This is the diacyl glycerol. One of the fatty acid chains is missing, and it has to create a hydroxyl group. Now, we're not going to talk about diacyl glycerol again probably this year, but next year, when we get to pharmacology and cell signaling, diacyl glycerol molecule is going to become a very important molecule through calcium signaling and activation of kinases. Here you have the diacyl glycerol. Now, it's the same thing. This is all the same thing as this, except this has a phosphate group. And notice this picks up two negative charges of the phosphate, and this is phosphatidic acid. And this thing is polar. All right. So now we're going to move on to more complicated lipid here. And we can start to talk about phospholipids. And the phosphatidic acid is the foundation of phospholipids. So what we can do is where one of those negative charges are on the phosphatidic acid, we can use that to put another head group. We can put another chemical moiety in place. And typically what will be done is you're going to put the most common one is phosphorylcholine. So this is your choline molecule, like acetylcholine. This is the choline. But you could also use ethanolamine or serine. And you take the serine or the ethanolamine or the choline and you conjugate it covalently to the phosphatidic acid. And you make a phospholipid. That is the definition of a phospholipid. Right? These things are going to be the substrate of bilayer membranes. And so we're going to talk about them for a while now. While we're at this slide, I just want to, you can see the difference in size between choline and ethanolamine. Notice first of all that they both carry a nitrogen. So they're both similar in structure. From the phosphate hydroxyl, you have carbon, carbon, nitrogen. And the only difference is that this nitrogen is basically an ammonia. And this thing is, has methyl group stuff all over it. So they both carry a positive charge, which will neutralize the negative charge on the phosphate. And so both phosphatidylcholine and ethanolamine are electrically neutral. They carry no net negative charge. All right. This thing is bulky. This thing is small. We're going to make use of that in a minute. Okay. These things are going to fit together. Phosphatidylserine has both a positive and negative. Of course, it's basically your amino acid, serine. And so there was a negative over here, there's a negative over here, there's a positive over here. So phosphatidylserine, unlike phosphatidylcholine and phosphatidylethanolamine, carries a net negative charge. Okay? If you put phosphatidylserine into a bilayer, the bilayer is going to have a surface charge that is negative. This is extremely important in the nervous system. These are space-occupying drawings from Leninger's book, which was a textbook written in 1969. I used this book during my PhD, so my tempo was a probe that I used to look at these the motion of these groups. At any rate, you can ignore that, but you can begin to see the size of the space. I want you to see the size. I want you to begin to visualize the membrane. I mean, when you live on the membrane, you have to see the sea of membrane that you're living on as proteins are approaching you. I mean, scale it up, make it macroscopic. See sitting on the membrane surface, okay? Ethanolamine is small, choline is large. Phosphatidylserine is roughly the same size as choline, but carries a net negative charge. And phosphatidylinositol is bigger than all of them, but also carries a net negative charge because the inositol is neutral. So the phosphatidic acid negative charge is not neutralized. So that's where it picks up its negative charge. All right. So these are our building blocks. Here's cardiolipin. I just put this in because I could. Cardiolipin is basically two phosphatidylethanolamines, phosphatidylcholines put together and carries a net negative charge because they interact. So you have a 4-chain fatty acid, 2 glycerols. That's what cardiolipin is. It looks a lot like phosphatidylcholine, but it's negatively charged. All right. That's sort of the structure of the basic lipids. Now let's start to put them together. Because they are amphipathic, that is, they have 2 acyl chains, they're going to be hydrophobic. But they have this big head group region now, including a phosphate group up in there. So this thing is going to be really polar. So this amphipathic nature causes them to align themselves to keep the head groups out into the water. The acyl chains get sequestered away. Their favored energy configuration of a phospholipid, not of a fatty acid anymore, but of a phospholipid, is an extended bilayer. You just take a bunch of egg yolk lecithin, throw it in the water, shake the water up, or put ultrasound energy in, and you get bilayers all over the place. That's what you do. And that's what my PhD was based on that model. Now, there's nothing covalent about these. These lipids are not covalently binding to one another. Now, think about the phosphatidylcholine, which had a positive and a negative charge. The head group is neutral, or ethanolamine, same thing. So it's overall neutral. But one end is positive and one end is negative. So now the ethanolamine, because it's flexible, it might just bend around and let its positive charge interact with its own negative charge. But the choline can't do that. So a bunch of cholines line up and the head group will come down from that positive charge at the choline and interact with the negative charge on the phosphate group. And this guy will do the same thing. And pretty soon you have an electrostatic interaction at the surface of the membrane. Nothing covalent, no bonds, but there's an electrostatic interaction. There's also very strong van der Waals forces of the fatty acyl chains because they all want to pack together. So there are a lot of forces that are going to keep this thing together, okay? But nothing's covalent. Therefore, because it's a semi-stable structure that has no covalent bond, it is known as a liquid crystal. And this is the paradigm liquid crystal. This is where the term liquid crystal comes from, is the bilayer membrane. Like that. And there's your bilayer. Here are your acyl chains. Here are your head groups. It could be, this is probably an inositol, but we don't really care. Choline, I mean, it makes no difference. The point is that we have this, see how this thing has bent over to interact with its neighbor? So you have very strong electrostatic interactions making this sheet of electric charge and hydrophilic material that is on either side. It makes a sandwich of a fatty lipid bilayer, okay? Now, this thing has some really interesting properties. First of all, let's look at the influence of the theta acyl chain. Now, if you remember from fat versus oil, if you put some really long hydrocarbon chains that are all just trans next, across from one another, those strong van der Waals interactions are going to lead to tight packing, and you're going to end up with a wax. Not very good for biology. We really don't want any wax in our, okay? So we're not, with maybe one exception, but we're not going to do that at body temp. We don't want that. Now, to make that lipid core liquid, you could either use a short acyl chain, or you can introduce cis bonds to break the structure. The problem, like coconut oil, is short acyl chains, C8, C10, C12. Those are not very good because there the van der Waals interactions are not strong enough and the membrane keeps coming apart. It will go back together again, but it keeps coming apart. It's not very stable. So to get to a stable bilayer, but which is liquid at the center, what you do is you use C16 and C18 acyl chains with cis bonds and interspersed. And that creates a nice, strong, stable interaction with a liquid core. And that's biology. That's what's all of our biologic membranes have that as a foundation. Okay? Like this. This is what we don't want. This is what we do want. Okay? Now there's another way to get to this that I'm going to show you in a minute, and that's really actually what my PhD was about. But first, let's get to this. So we talked about the influence of the acyl chains. Here's the influence of the head groups on the structure of the bilayer. First of all, we've already been over this. Phosphatidylcholine, phosphatidylethylamine are neutral. Phosphatidylcholine is bulky, phosphatidylethylamine is not. Now, if you wanted a curved membrane, don't you love it? Just put phosphatidylcholine on the outside, phosphatidylethylamine on the inside, and it's going to curve. Well, won't the molecules, won't they randomize? Won't the ethanolamine flip-flop? The term is flip-flop from the inner bilayer to the outer bilayer. And the answer is no. I'm going to show you that in a minute. So this is going to be a stable, once you make the membrane, there's no exchange of the lipid from the inner leaflet to the outer leaflet. It's forever, basically. Very interesting. Okay. Phosphatidylserine and phosphatidylethanolamine are big and they carry a negative charge. So if you want to stick a negative protein, excuse me, if you want to stick a cationic protein onto the surface, you want to expose some negative charge, make a negative charge sheet, and something like myelin basic protein, just simply lay right on it as an electrostatic interaction. There are tons of molecules that do that, and sometimes you want to shield, and for instance, in the endothelium, which is filled with negative charge, phosphatidylserine, in order to avoid immune responses against you, in order to prevent a vasculitis, you have to shield those negative charges. And lupus, for instance, is a defect in the shielding protein, so that the phosphatidylserine is exposed, and you start a vasculitis that way. Okay, so these kind of electrostatic interactions, we're going to get to these when we look at proteins and how they interact with the lipid. And of course, water is of critical importance because water has a high dielectric constant. Now that's going to mean something. In other words, it can orient itself toward a charge. Hydrogen and oxygen are very different from one another. So the oxygen can orient itself toward a charge if it wants. And so when it does that, a negative charge, an electrostatic surface, will orient water and actually create water groups that are usually about 9 water molecules as a structure. So the water is not just floating around free. And now this is what the yellow and white is the water. So now our bilayer is getting more complicated. We haven't added any proteins. We haven't done anything with the nature of the phospholipid head groups yet. Okay, we're just looking at it generically, and it's already got a lot of structure to it. Okay. Now this is where my PhD actually was what happens when you add cholesterol to this bilayer? What's its effect on the head group motion, and what's its effect on the acyl chains? So the main finding was that when you add cholesterol into this bilayer, so first of all, cholesterol is a block. It isn't flexible at all. It's a very complex structure and a fairly large structure. So it's a rigid planar ring. It has a bulky tail and it has a hydroxyl head group. And when you drop cholesterol into a bilayer, so it goes into the lipid section, the hydroxyl group will orient it toward the water. But what it does is, it's only, as you're going to see in a minute, because it's shorter than the acyl chain, it interacts with the acyl chain close to the head group, and then it isn't there where the tail of the acyl chains are. So that's what it's going to do. It's going to put the membrane in an intermediate state of fluidity. Now that's really important because you want your membrane resistant to temperature changes. You want your membrane resistant to forces, structural forces. And the amount of cholesterol varies. It can be anywhere from none to 1:1. So here's your cholesterol molecule, rather short chain on a rigid rings plane, like that. And there's your space occupying model. And here you are. So here's a fatty phosphatidylcholine, and here's your cholesterol in between. Okay, and see, so this is going to pack this section fairly tight. It's going to restrict the motion of this section, and these things are going to be whipping around. Now, I meant to tell you, when you just go back for a second, when you look at these chains and you see them static, what you really need to do is think of this thing as a whip. Okay, it's moving like a whip. It's also, okay, precessing. So it's turning, okay, it's precessing, it's whipping, okay, and it's moving in general about 16 billion times a second. So 16 billion times a second. So when we say that it's liquid, we mean it's liquid, okay? This thing is normally moving really, really fast. And so is this. And all these things are just, all of them are whipping. Each chain is going at 16 billion times a second, all right? So when you put the cholesterol in there, that's going to slow this whole thing down and give some structure to this upper section. But look at the space that it creates down here. And these things go nuts. They just go nuts, okay? They just start moving around at high speed because there's nothing restricting them whatsoever. They don't have any polar, nothing polar in there. It's all acyl. And so they're going to intercalate with one another. They're just going to, it's going to be an oil in the center, which means as an oil in the center, it means that anything polar that wants to get through the bilayer is going to have a hard time. Anything nonpolar isn't even going to be able to get to the bilayer because it's got to get through the head region, and anything polar will not cross the bilayer. And there starts to, now you see how the properties of the bilayer are emerging from the chemical structure of the phospholipids. But we want to make it more complicated. That's not very interesting. That's just the native property of this spontaneous structure that occurs. All right. These are the basic properties. It's stable. It reanneals if you disrupt it, but it's very stable. Our biologic membranes are very stable. The thickness is usually about 60 angstroms, approximately 6 nanometers. All right. The electrical resistance can be studied and is very high. The electrical resistance to these things is very high. And it classically has been studied. You take the phospholipid, you paint it onto a pinhole, and then you can study anything you want. You can study transport, you can study, and you can change the composition of your membrane and watch things go through. All right. We're still with the basic bilayer, all right? So let's talk some properties I've alluded to. First of all, if something wants to go across the membrane, first of all, so anything, any molecule, let's say glucose, all right, and you could be urea, it could be anything you want, is going to have a water shell when it's in solution. It has to get rid of the water shell. It has to enter the hydration shell of the membrane. Then it has to go through the hydrophobic region. It's going to diffuse across. Then it has to get out into the polar region, rehydrate itself, and get out into the cytoplasm. So multi steps. The rate limiting step is the diffusion across the bilayer because it has to get through that lipid core. Therefore, the permeability coefficient of any small molecule through a bilayer will correlate with its lipid solubility. It can overcome everything else. It cannot get through the hydrophobic region if it's polar. So look at these. So water is special. Water gets across the membrane with a half-life of 1,000th of a second. Urea, 1/7 of a second, okay, for urea to go across. Glucose, 24 seconds for glucose to cross a bilayer on average. Chloride, a day. You take a chloride molecule, go back to this, put a chloride molecule over here, wait for it to get over here, it'll take a day. Okay, fill the chamber with chloride. Only half of it will cross in a day. All right, that's what we're saying. Potassium, 20 days. Okay, sodium, 2.5 months. The membrane isn't letting these guys through. So if you pump charge, it's done. It's stored on the other side. It will not, the battery doesn't degrade spontaneously because of these properties. Okay? If you're going to move charge, then you're going to have to move them through channels. The charge will not move across the bilayer by itself. The other thing is what I call flip-flop, where one phospholipid will move from one leaflet into the other leaflet. It is basically forbidden. Okay, it's forbidden. It just simply doesn't happen. It would take months if you labeled a phospholipid on one side of the bilayer. It'll be months before that label appears on the other side of the bilayer. It basically doesn't happen. In biology, this asymmetry is created in the endoplasmic reticulum. So if I said, where are membranes created? Where's the cell membrane created? Where are synaptic vesicles created? All of those things. Where's the Golgi? All of it is done in the endoplasmic reticulum, right? And you think of the endoplasmic reticulum as a sac in which something's happening. Wrong, okay? The endoplasmic reticulum membrane is a very active part of this. So it's doing the synthesis. It's putting proteins on the inside membrane, not the outside membrane of the endoplasmic reticulum. So the inside of the endoplasmic reticulum, you have to think of it as away from the cytoplasm. Don't think of it as inside the ER. Think of it as outside the cytoplasm. So let's say that there was a protein or a lipid that you really didn't want to expose the inside of the cell to. It's destined to the outside. It's going to be, I'm going to make a cell receptor. Think of a cell receptor, right? I'm going to make the cell receptor in the endoplasmic reticulum. Typical cell receptor is going to be a glycoprotein. So I need to put the glucose molecules, whatever the sugars are. Well, I do that in the Golgi apparatus. So I maintain the same topography as I had in the endoplasmic reticulum. It's away from the cytoplasm, in the Golgi. Then I bud off a little piece with the receptor in it. It's on the inside. The cytoplasm doesn't see it. And then I move my vesicle to the protein, and I let the vesicle fuse with the membrane in such a way that my receptor was never exposed to the cytoplasm. That's where we're going. That's sort of practically the end of this talk. Okay, that's where we need to get to. Okay. Despite the fact that flip-flop is not allowed, lateral diffusion is unbelievably fast, and a molecule could get from one side of a cell to another side of the cell in less than a second. So this direction is whipping through, and this direction is basically behaving like a liquid. And again, you have this liquid crystal, this peculiar array, all right? And the diffusion occurs within the monolayer, never crossing, no flip-flop. The other thing that it took us a long time to recognize is that even lipids alone, but once you start adding proteins, lipids and proteins form groups like a receptor, and they can travel, okay, in the bilayer, in one side of the bilayer. Again, they won't cross, they won't flip-flop either. That is known as a raft, which is a cool name for it, because you have a bunch of lipids and the proteins that are traveling as a unit, and so that's given the name raft. And this is, so now we're getting into more complex structures where you can ignore all this for the moment, we'll come back to it. This is nice, here's your head groups, here's the cholesterol, and here's a raft. And the raft can travel very quickly. And it could be dragged, by the way. We'll see how it can interact with other parts of the cell. All right, take a break from this structure, and let's go back to lipids themselves. So in the nervous system, you have a lot of sphingolipids. So let's just talk briefly about what is a sphingolipid. A sphingolipid is similar to a phospholipid, except its backbone is not glycerol. Okay? That's the main difference, and I'll show you that in a minute. It's a sphingosine. Okay? The fatty acids and sphingolipids in the nervous system tend to be very long, and they also tend to become unprotonated, and so sphingolipids often carry a charge. And here you go. So here's phosphatidylcholine, so you're familiar with that. And here's the sphingomyelin, which has a choline head group, so it looks a lot like phosphatidylcholine, but the backbone, which was glycerol, is not. It's sphingosine, and there's a nitrogen group in there. And that makes it more polar than it was. But and also you have these very long, look how long this is. I think even with these models, you can see how long this chain gets. All right. And here's sphingomyelin, and here's phosphatidylcholine. And they look similar, but you can see the nitrogen group. However, the sphingolipids are typically not left alone as sphingolipids, and the sphingolipids form the glycophospholipids. All glycophospholipids are sphingolipids except for phosphatidylinositol. Phosphatidylinositol is a phospholipid. All other glycolipids are sphingolipids. Okay? So all of the neurominic acid, those sorts of things that we're going to be talking about, those are all on sphingomyelin. All right? They are virtually only on the external surface of the plasma membrane. You virtually never have any of this pointing into the cell. All right? And we talked about that already. Glycolipids make up about 5% of plasma membrane lipids, including in red cell and most cells. They can reach much higher levels in the neurons. and especially in the region of the synapse. Right? They are very effective at creating shielding charges. They are wonderful for cell recognition. They also, unfortunately, are good for autoimmune attack, and they provide receptor specificity. And here's your structural. These are big head groups now. Now you can see it's a glycolipid. These head groups, this might be phosphatidylcholine, might look like that. So these are, and this is only maybe 2 sugars. You can have 5 sugars on them. And so the head groups can be really big. And here's an old biochemistry, this is from the 60s, when people were first studying separating lipids and TLC and that sort of thing, and measuring lipid quantities. And you can see, if we look at myelin, that like the erythrocyte, it has a lot of cholesterol, but unlike virtually anything else, it has huge amounts of glycolipid. All right. So myelin has a lot of glycolipid, negative charge, myelin basic protein, tightly adherent membranes, very stable, nearly wax-like structures, long chain fatty acids. The structure, the chemical composition of these things goes with the function, okay? If you want a different function, if you want a stable membrane that isn't going to let much through and isn't going to allow any charge through and anything like that, well, then what you want is an insulation. You just want it, doesn't need to be mobile, nothing's crossing it. Keep the water the hell out of it, okay? Just make a waxy structure that is tightly adherent, layer upon layer upon layer. And that's what myelin is, and it's chemical, very little phosphatidylcholine, okay? Now we really got to make this, add some serious function to this. Right? So the protein content can vary. Myelin has very little. It's not doing very much except sticking to itself. All right. Mitochondria are all about charge and energy transport and that sort of thing. They're 75% protein. They have very little lipid. Those are your 2 extremes. Typically, it's roughly 50-50. Yep, sure.
Does our lipids like cholesterol and triglycerides and all that, does it have to do anything with the lipids that are inside the brain cells? Like having lower cholesterol levels, is that neuroprotective for cell, like neurons
So
for
probably
their
not.
metabolism?
Probably not at the neuron level. So if you're saying, can you alter the cholesterol content of myelin through diet? The answer is probably not. It's going to do what it's going to do. After all, the brain can make its own, the body can make its cholesterol. In most cases, the brain can make its own cholesterol if it wants. However, if you lower cholesterol, typically into the below 165 milligram percent, into the 130 range, the blood vessels, you're going to change the cholesterol content of the membrane of the blood vessels. And that is probably one of the reasons that a low cholesterol is associated with a high risk of intracerebral hemorrhage. Remember, brain arteries have a wall thickness that is only 2/3 that of every other artery in the body. And the posterior circulation is even worse.
And what about with, let's say, memory, like the metabolism of the cell?
In general, the brain is going to regulate its own chemical structures, and except in the cases of extreme deficiency, it'll make all the glycerol it wants. It can certainly make all the sphingosine that it wants, and it will, unless you just have extraordinary dietary deficiencies, really kwashiorkor level deficiencies, it will deal with itself. That's why if you look at kwashiorkor, the child's head is so large because the whole body just diverts all of its energy and metabolism to preserving the brain. It's the last thing to go. I think we've covered all this. Now, I'm just going over the varieties of interactions that lipids can have with protein, and we're going to be dealing with this over the next 2 years. When we get to the synapse, you're going to want to understand, vesicle fusion, right? You're going to understand the whole synaptic mechanism. It's necessary to have this in order to understand how the synapse works, okay? And we will get to that this year, I believe. So these protein interactions with the lipid bilayer can be hydrophilic, so non-covalent, hydrophobic, that is, they can embed themselves in the lipid core if they want to. They can just simply sit on the surface via an electrostatic interaction. They can be completely integral, having charges at either end of the protein and a big lipid alpha helix in the center. And I'll show you pictures of all these. Sometimes the proteins themselves do not enter the bilayer, but anchor themselves by having a phospholipid as part of the protein. So you have a lipoprotein. And the lipoprotein can embed itself into the membrane, and the protein sitting out there, never entering the membrane. As a general rule, no proteins terminate within the bilayer. Indeed, how could they? Because you're going to have a carboxyl group or an amino group, and that will not stay in the hydrophobic core. It's going to pop right out, right? Because you have a charge in the acyl, and it can't live in that acyl domain. So as we get further into more complex membranes and start to look at the biology of membrane, and indeed all of macromolecular chemistry, it begins to fuse with electron microscopy. So you can actually see and visualize these macromolecules. And so here's just the first of them, where you're going to see a lot of them from now on. And here's a bilayer. You can see, first of all, you can see the lipid bilayer, which is this sort of semi-lucent section in the center. You can see proteins laying on the bilayer. That's the dense core. And on the external side, these things go way out, like the corona of the sun in the eclipse. They go way out into the extracellular space. You don't see a whole lot of that on the intracellular side. Cartoon, nice, explaining, here's some examples of how proteins can interact. They can be amphipathic. This is an alpha helix. The alpha helix has, as the alpha helix forms, you line up a bunch of polar amino acids, you line up a bunch of non-polar amino acids, and you basically are creating a rod in which part of it is going to orient itself. The charge is going to head toward the surface. Okay, the non-polar section is going to want to embed itself in the bilayer, right? And so you end up with this alpha helix, which is then connected to the rest of the protein chain, all right? Or you can have strictly hydrophobic helix coils, in which case they just embed themselves in the membrane. Or you can attach yourself with a lipid anchor, or you can have electrostatic charge interactions like myelin-basic protein would do. So these are just cartoons showing you what I've been saying. Here are molecules now which span the entire membrane. Okay. Not all of them will. Here's examples of a protein embedded in you can see the amino linkage, and here's a fatty acid chain. Here's it's stuck with a cholesterol molecule, a phenyl group. Here's an alpha helix running right through. So now this protein, which would be an integral membrane protein, is exposed to both surfaces. All right, and this keeps it embedded in the bilayer. And here's just the same thing. None of these are polar. All of these are non-polar amino acids, and so it's basically hydrophobic, and it embeds itself in the membrane. That's a transmembrane protein. And here's just a summary of all the different ways that you can do this. Okay. Now, we've already said that proteins can interact with specific phospholipids or other lipids, glycolipids, or anything that it wants, and you can create a raft, and the raft travels around as a unit. Right? Think receptor if you want, okay? But it doesn't have to be a receptor, but that's sort of the easiest one to think about. The lipids don't flip-flop, the proteins don't flip-flop, period. So any asymmetry which is created in the endoplasmic reticulum in the Golgi is forever. Okay, it's forever, basically, for the life of the cell. You can have a variable lateral diffusion. If the protein is sort of floating in the sea, it can diffuse almost as fast as a lipid can diffuse. You get from one side of the cell to the other in a second. Okay? If it's interacting with stuff on the inner surface of the membrane, and it's interacting with something underneath the membrane, it ain't going nowhere. It's going to stay put for as long as the cell wants it to, until the cell now could control it. It can move it if it wants. It can release it if it wants. It can do all sorts of things. Yeah, think about it. And so this is just now you can start to build these things up. Here's an adsorbed glycoprotein. So here's a protein which never is embedded in the membrane at all. This thing is an embedding protein. It may not have any function at all. Protein kinase C is a good example. Okay, protein kinase C doesn't really enter the membrane. It's a protein that is held in place by other proteins which are embedded in the membrane. Okay, so that sort of thing goes on all the time. And fuzz, sometimes this is referred to as fuzz. Okay, so you can see the orientation. Here's the nucleus. This is extracellular, and you can see how far the membrane proteins extend, but not on the inner surface. Some, but nowhere near like it is on the external surface. And our picture of graphs and now proteins, and halfway across an electrostatic and peripheral proteins, and you start to see a summary of everything that's going on. What you do with these proteins, you transduce energy. Energy, okay, energy transduction. In other words, muscle, motion, okay? Convert it into mechanical energy, if you like, all right? Or you can transport ions, transport proteins, transport anything you want. Create ion channels that are variable that you can control the flux of the sodium or potassium or the chloride or anything else you want, because from a practical point of view, they cannot cross the bilayer. Okay, they're stuck. Make a disequilibrium, it's more or less permanent until you use it. Okay, receptors. Okay, activation of channels, cell recognition, intercellular, everything that you're going to be thinking about over the next 2 years in terms of physiology, nearly all of it is occurring on the membrane surface. Even what you think of as cytoplasmic proteins, and you think they're not interacting, they are interacting. They're just on the cytoplasmic surface of some organelle. There's very little, but very little, that's floating free in cytoplasm. Cytoplasm is a highly structured environment, which is why, if you think about diffusion weighted imaging, remember, just think, so we're measuring diffusion. Diffusion in the extracellular space is very fast because it's unstructured. When the water molecule goes into the cell, why does the diffusion fall to 20% of what it was or less? It's because it's in a structured environment. It's in a gel, okay? Don't think of the cytoplasm as a bag of water. That won't work. That's not the way it is. Because otherwise, you're not going to be able to move a small molecule, let's say from the mitochondria to the cell surface. How are you going to, you're going to wait for it to diffuse, to get there by chance? The cell's not going to wait for it to get there by chance. The cell is going to move it along a transport system, which means that there is a structure inside the cytoplasm. And all of it is interacting with the cell, with not only the cell membrane, but the membranes of the intracellular organelles. Okay, you have to think of the cell this way. All right. All right, now, and that's basically where we're going, is we're talking about how do we make domains. So why, some of the stuff, dendrite is a domain. Well, you say, well, what makes a dendrite a dendrite? Well, it has, come on, it has a cytoskeletal system. We'll talk soon about cytoskeletal system as part of our macromolecular chemistry, and okay. But it's interacting with the membrane. If it doesn't poke a hole through the membrane, does it? Okay. If so, somehow the membrane is attached to the cytoskeletal system, so you can change the shape and make it longer and put bulbs at the end of it for synaptic terminals and all this stuff. Transport molecules through it. All that has to happen. Sometimes, like in the case of the muscle, okay, you have to convert it for mechanical energy. Or in the case of the synapse, you're going to have to deal with exo and endocytosis. All of these things are going to have to be explained by the macromolecular structure of the membranes and their interactions with the rest of the cell. And so it's not alone. So here's a great example. This is from a red blood cell. Here's an anion channel on the surface of the red blood cell, it's known as AN3, carrying negative charge. Lots of hydrophobic side chains, fully embedded, making a channel, fully embedded in the bilayer, but not just loose in the bilayer, but interacting with the actomyosin skeleton of the red blood cell through a series of proteins called anchorin and spectrin. You can move, the red cell can move these things around in response. The platelet is going to do that, right? And the neuron is going to do that as well. And you start to get structures now that look like this, where you have cytoskeletal elements that are underlying the membrane and can move things around and create receptors that it can hold together. Move a G protein, right, in association with an ion channel. If you're waiting for the G protein and the ion channel to bump into one another and adhere, just hit each other just right to adhere to one another, it is never going to happen, okay? Come over here. You know, I come over here. I want to talk to this other cell. So get your butt over here. Come on over here. Interact with one another. Expose yourself and have an interaction with the next cell. That's the kind of thing that you have to imagine. And here's the EM. This is not just some cartoon rendition of how these things work. This is the EM of how these things work. They're there. They're always like that. Okay? And that's it for today. And that's just the first building block.
Did you say brain?
Membranes.
I say the same thing.
I borrowed a term from cosmology.